All right, good. So I was actually just going to quickly recap for anybody who was not here last time. I thought we could quickly just run through the presentation from last time and see if anybody had any questions. Okay. I think we started off with asking some questions regarding, what medicines have people usually taken. We gave these examples usually, right? Crocin, Benadryl, Gelusil, etc., for different things. And we started speaking about, what is in these medicines, right? So we spoke about how, for example, Lay's, which is potato chips, usually has potatoes and vegetable oil, right, as the main ingredients. And we started talking about, well, if this is the simple ingredients in Lay's potato chips, what is the more complicated ingredients in something like crocin or Tylenol? And we spoke about how the active ingredient is the one in the medicine that actually helps cure the disease or the problem, and the inactive ingredients, right? Those are the things that are in there just to help with making either the right shape or the texture, or to maybe give it some more stability at temperature, making sure that it doesn't go bad or anything like that. Then we spoke about a little bit about the history, right? That long ago, in ancient times, medicine and religion were very closely intertwined with each other. Might not necessarily be the best thing, because some of these cures weren't scientifically accurate, scientifically researched. These were just religious. And we spoke about how some of these medicines obviously are still good, did work well for everybody, but some medicines did not work for everybody. So I gave this example as Ficato. It is one of the medicines that was gotten from a milkweed, which was an old effective medication. This was passed on from generation to generation until it got into the lab of a scientist who was actually able to procure it and make sure that it can actually cure diseases the right way. We spoke about the first person, whoever actually developed the medicine chemically, and he made it in the lab. What he made was he made morphine, which he made from opium after he realized that everybody who eats opium feels sleepy all of a sudden. And then you can see that, wait a minute, maybe there is something in the opium that can help people sleep. So he extracted the morphine from the opium, and then when he was doing that, he tried to test it on his friends, and then we spoke about how that cannot be done anymore, right? Because there are more rules and regulations around medicine making. And we spoke about how so many years of science and effort is usually required to take a develop a medicine right now. And we ran through different stages of drug development, research, development, manufacture, what they all mean, and why each stage is important and takes time. And then we also spoke about how we usually test the medicines, right, in different stages. We do animal testing, we do clinical trial testing for all the medicines we do in different phases, and that some of them are most of them are very difficult to succeed, and how it takes a lot of time and a lot of money to be able to do clinical trials, but it is still difficult for us to still develop new medicines, just because it is very complicated, because there are a lot of steps involved here. And then it is actually a very difficult thing to do. So that's why it takes about 6 to 8 years, and $1.3 billion, which is a lot of money, right? And next, we spoke about how everything all the chemicals and all the drugs that we spoke about are actually chemical drugs. The chemical reaction that you see on the right here is what is used to make acetaminophen, which is the most common, drug for cold, fever, etc. And we started talking about how chemical drugs have been there for a long time now, but unfortunately or fortunately, they are easier to make. But the problem is that they don't cure all the diseases we want. The complicated diseases, the more problematic diseases, it seems like the chemical drugs cannot cure them. So we started talking about, well, as the modern medicine field evolved. We have to find a new way and a new type of medicine to be able to cure the more complicated medicines. And this is by making therapeutic proteins. So we just started speaking about proteins. I asked, proteins. When what we mostly talk about proteins when we talk about food, right? So some people gave me suggestions saying that, they hear about it when they talk about nutritional supplements, all that. Absolutely correct. Right, but so today we will talk about, from this point, we will talk about, well, proteins. What do they mean? What type of proteins are there? Why is it important? And, how do we make these protein-based medicines? So now, I guess I'm going to start a quick funny question for all of you, right? There are two people here in this two photos, right? Tell me, who do you think has more weight? Who do you think weighs more? Who do you think is heavier amongst these two? You can guess. Between these two people on the screen, who do you think is heavier? Who do you think has more weight?
First person.
First
First
person.
person? Okay. You mean the person on the left? Right? This guy? Okay. This guy, right? Okay. So let's look at their weights. They're actually the exact same weight. Okay. So they're actually the same weight. Now, can someone tell me why they look so different? The doctor always says, right? Keep yourself healthy, keep yourself, make sure that you don't put on weight. But these two people, they're the same weight. Why do they look so different?
So because their muscles are shaped like different.
Okay, correct. Yes, muscles are shaped, of course. But this person, does it look like it has too many muscles?
So he has more fat.
Yeah, right, correct. So, when people keep talking about, that you should lose weight or you're too heavy or anything like that, no, right? It doesn't matter. Like, what is more important is, why you're heavy, right? So clearly this person is healthier, right? Whoever the rock is much healthier than that person in this picture here. And why is that? Well, why do they look different? Well, you can see here, right? So 2 kg of muscle, right, occupies much lesser space than 2 kg of fat, right? So if your body has more fat, then you automatically start looking bigger, right? But if you have muscle, then you look smaller, even if you have the same weight of the muscle or the fat in the body, right? But clearly one of this is healthier, right? We don't necessarily like fat. Our body likes to accumulate fat, but fat isn't usually good for you or good for anybody. So that is why whenever we start focusing on healthier things, we say, well, this is healthy and this is not healthy, right? So how do you do this? Well, you eat the right thing, right? And whenever they talk about eating the right thing to grow muscle, they always say eat protein, right? So that is why protein, easiest thing to talk about. It is most commonly talked about when we talk about food. And yes, he's absolutely a fit person. And then yeah, so this is example, right? Two people who weigh the exact same amount, the different levels of health because of the body composition. The fact that muscles itself are actually made of protein. So you can only develop this if you eat protein. So that is one of another roles of the way that protein plays in the body, right? So this is another reason why it is very important when we talk about proteins to understand that they play different roles for different people, that it is not one thing or just, a very small part of it. Yeah, proteins have multiple roles in the body. So, right, so this is what we're talking about, right? Proteins are considered to be healthier than fats, right? And why do they be healthier? Well, unlike fat, right? Unlike fat or any maybe even some carbohydrates, they don't have too many negative effects, because everything that you eat, every protein, if you like, not too much protein, but as long as you eat enough protein that your body needs, usually what happens is that it goes into your body and it starts accumulating as muscle, right? So even if you don't tone your muscle, right, even if you don't go to the gym, your body still has the muscle, right? So that is definitely much better than eating the fat, because fat will have other problems in the blood. It'll clog your heart. It might cause your it will deposit around other organs of yours and not let it function properly. But protein usually does not have these particularly bad connotations. So this is the reason why, we have so many benefits. And when we talk about eating protein and all these people, bodybuilders who eat protein, this is why, because they can eat the protein, it helps them develop muscle and doesn't have too many other negative effects. So in terms of food, apart from protein supplements, what are the things that already that has lots of protein? Can you guys give me some examples of maybe what has proteins?
Milk,
Correct. Milk. Yes, absolutely.
egg,
Correct. Egg also.
meat,
Brilliant. Yes,
wheat,
correct. So if you are vegetarian, what do
So
you do?
meat,
You said meat. Yes, yes, correct. Absolutely. So if you're vegetarian, where do you get your protein from?
so dairy products like milk and curd and cheese.
Correct. Anything else? Any vegetarian sources of protein?
So mushroom.
Mm, yes, but not lots of protein. I think it will have
soya,
some protein. Yes.
underground plants,
Correct, correct. Yes, all these green gram, yes, all these pulses, right? Dal, rajma, chana, green dal, right? These are all things, legumes, exactly, are what has protein. So why do legumes usually have protein, right? Because I'll go to the next slide and we will talk about what proteins actually are, right? So in terms of chemically speaking, like what are proteins, right? Proteins are peptides and they're polypeptides. What does that necessarily mean? Well, a peptide is think of an amino acid, right? So we all know that COOH is an acid, right? Amino means that they had nitrogen, NH2, right? So this is an amino acid. What does they mean by polypeptide? Well, polypeptide simply means that there are two amino acids coming together, right? So again, pure easy chemical reaction, right? So there is OH here, there is H here, it comes out as water, and then these two come together and form out one peptide. So this is itself is a polypeptide. What does polypeptide means? Two, multiple peptides, right? In this case, there are two. So imagine a protein, right? This is just two. Proteins just have much more of these peptide bonds put together. So there are some proteins which are so large, it has like 150 or so peptide amino acids coming together to form a peptide bond. Now, you spoke about green gram, legumes, all those stuff, right? Well, how do they have more protein than not other things? Well, if you remember from school, right, all these legumes are all these underground growing like dal and green gram and stuff. How they usually grow is that they grow underground, right? So they have bacteria that will help them get nitrogen from the atmosphere. And you see the nitrogen here. So that is why it is more easy for you to find protein in this, because they're one of those few bacteria which can actually get the nitrogen from the atmosphere into the mud. And that is how we can grow green grams and legumes, and they will still have protein. Not as much protein as meat, but they definitely will have more protein than most other vegetables that grow over the ground. Anybody remember the names of those bacteria that help get the nitrogen from the Earth's atmosphere?
So rhizobium.
rhizobium,
Correct, absolutely. So this is an example, right, of all the amino acids examples. So there are, as I told you, when they come together to form a polypeptide, um, we a lot of them coming together forms a one big protein unit, right? But what are these amino acids itself, right? So there are lots of types of amino acids as well, but these are the ones that are naturally occurring in the human body, right? All these amino acids, yeah, they occur in the human body themselves on their own, right? So we don't necessarily have to consume them, right? It is already in the body. So think of all these combinations, right? So for example, each of them has different things, and there are only few with sulfur, right? So if you look at the yellow color here, there is only one here, methionine, which has sulfur. But everything else is pretty much what I showed you, right? There is nitrogen. There's always has to be at least one or two nitrogens. Most of it is carbon. There will be some hydrogens, and there has to be an oxygen, right? Because this acid is COOH, right? So it has to have at least one oxygen and some hydrogen to form an acid. So I'm just trying to show you here, like what is the like almost minimal type of amino acid combinations, right? And now you can already see that some of these structures starting to become complicated already, right? You have so many little branches here, you have so many little branches here, and this is just, some amino acids, right? Now, if they have to put more and more of these together in different combinations to make a protein. So think of how complicated a protein will be, right? At the end of it, think of 100 of these put together in different shapes and different forms. You can put them together like this, you can put them together like this. It ideally becomes very complicated, right? So that is also the reason behind the complexity of, if we had to make a protein in the lab, right? It suddenly becomes more complicated, right? Because I showed you in the, remember the earlier slides, right? To make the acetaminophen, the chemical reaction which we needed to make that, at the end of it, it took 6 steps, right? It took 6 steps to get to the final product almost. But for a protein, that is not enough. Well, why? Because each of these branches now, right, it is more complicated. And because the starting material itself is actually so large and so of different types, that it becomes fundamentally more difficult to actually create something of these two components. So I'm going to, again, standard, right? What are these proteins and how do they help us, all right? And very simply, they have different roles. Again, this is not, the point is to not to, read this or learn this or by heart this. We're just trying to understand here, how do they affect us, right? In a big manner. So we already spoke about muscle, right? And then these are specific proteins, right? We like actin, myosin. These are all things that help build muscle, right? And so these are the reasons why the proteins have so many different ways or so many different requirements, then that it plays a massive role in everything. But I guess for the next couple of parts of this talk, we will focus on the proteins in the immune system, right? It's focused on antibodies and cytokines, but mainly antibodies, and we will talk about more of those. But quick examples, can anybody give me examples for anything, like a hormonal protein? Can someone give me an example for, hormonal protein? A hormone have, what hormones do you think we have? Example of a name. Any of you remember anything that was taught in school?
Insulin.
Correct. Enzymes? Remember the names of any enzymes?
For carboxypeptidase.
Absolutely correct. What does carboxypeptidase do, Roshmita? Anyone?
Some
Any example?
conversions between peptones, dipeptides.
Correct, right? So absolutely. So you just saw that we saw in the previous slide of what these proteins are like, right? But our body cannot actually digest them without breaking them down, right? So these enzymes like carboxypeptidase are responsible for breaking the bonds, right? So that we can actually, our body can start digesting these things. Our body can't digest any complex molecule, right? Anything that we eat, it has to be broken down into smaller pieces. So our body cannot consume any of this on its way completely, right? It has to be broken down into, smaller chunks, and that is how enzymes usually help. All right. Any last protein that you guys want to tell me about that you know? Anything that ends with an ase, right? So peptidase, sialidase, all these things, the name usually resembles that of an enzyme. So thyroid is a hormone, right? I'm sure all of you have heard of thyroid, right? That's a hormonal protein, right? Right. So moving on, we'll get into proteins in the immune system mainly, right? Because that is what we traditionally use when we want to make medicines. So there are two classes. One is of two types of protein in the immune system. One is the very quick thing. We'll just talk about it for 2 minutes, and then we'll go to the main one, which is the antibody. So the first type is the cytokine. Well, why is this important, and how is the cytokine important? Well, when your body thinks that it is falling sick, or that there is an external source by like a virus or a bacteria or even something in the body, right? Our body needs to know that there is a problem, right? Our body needs to react so that the other cells or the red blood cells or the white blood cells can actually trigger an immune response, right? Can they create our own antibodies? But something needs to know, right? So our body needs to understand it. So this is where this class of molecules called the cytokines are very useful. What do they do? Well, is that whenever they think that the body is going through any trouble, or that the body is being attacked by internal or external sources, they start doing these things. They start releasing these chemicals to other parts of the body to help them fight the disease or fight anything that is going on. So these are called cell signaling. So they send signals to different parts of the body, right? So you can see what all they need. They take information from lymphocytes, macrophages, so on and so forth. They accumulate all this information, and based off of this information, they will send signals to different parts of the body. Why do they need information from all these types of cells? Because the cytokines need to know, right? There is each time it'll look at the the balance of the lymphocyte. It will look at, you've heard of pH, right? It will look at the pH of the lymphocyte. It will look at the size of the macrophages. If any of these things start going away from normal, then the cytokines think that wait, there's a part of my body which has an issue. I'm falling sick. It might not work as like my body might not be working as well. So then communicates to other parts of the body, right? The more other parts of the immune system which can create antibodies can actively protect our body, right? So that is the main role of the cytokine. It is to send signals. But what does it send signals to, and what happens to the body after that, right? Any sort of defense in our body has to be done by, I'm sure all of you have heard, right? It is called an antibody, and which will come to the next slide. But I have a question here. When we spoke about this little bit last week, and we spoke about how chemicals have different, we make different chemicals for different diseases, right? But we also spoke a little bit about one of the things that we do to make sure that it is safe is because we make sure that there are no bad side effects. But I am sure that all of us, when you have eaten, any crocin or Benadryl or anything like that, right? Just take an example of Benadryl. Whenever I drink Benadryl, right, I feel sleepy. Everybody feels sleepy. Why is that? It's a side effect, right? Why do you think that side effects are caused by medicines that you take? Why not? Why does it cause any other effect apart from just helping you cure your cough or your cold? Why do you feel sleepy, you think, when we drink Benadryl? Questions, answers, thoughts, suggestions? Why do some medicines give us side effects? Why do people feel sleepy after drinking Benadryl?
Reaction with uh, our body parts and
Correct. Yes, it is. It is definitely a part of the reaction with the body, right? Anyone else have anything to say? So the side effects, right? They're usually because it is much more complicated, right? So developing a medicine, or let's say even for something like cold, right? It isn't necessarily easy. There isn't one part of the body that causes cold and can cure cold, right? There are multiple parts of the body involved in a cold. So one of the things that happens in a cold is that there is something in your body called histamine. Now, histamine is a type of hormone. It's a type of protein in the body that helps you react to a cold. So this is what says, you know what, I have a cold, I will cough, I will sneeze so that I can expel some of the germs out of my body. So that is the job of histamines in your body. Now, what Benadryl usually does is that whenever you drink Benadryl, it suppresses the histamines, which basically says Benadryl goes into your body and says, don't produce any more histamine, right? And that is why you don't cough as much or sneeze as much or have a cold as much when you drink Benadryl, right? So that helps you feel better about yourself when you have a cold. But the problem is also that histamine is what is responsible for keeping us awake. So when we wake up from sleep, histamines are what is responsible for for waking you up after you sleep. There are different hormones, of course, also, but histamines are very, very important in making sure that we are awake, that we are alert, we are not sleepy, and then we can, go on day-to-day activities for ourselves. So now do you see what happened? That the histamines is what causes your cold. Doesn't necessarily cause your cold, but causes the side effects of the cold, right? Coughing, sneezing, etc. And Benadryl helps with that. But also histamines are responsible for keeping you awake. So when the Benadryl usually hits the histamines, you suddenly start feeling sleepy because what is responsible for keeping you awake is not happening anymore. So this is an example, right? Why it is so complicated to develop a medicine, right? Because all these proteins, right? All these cell signaling molecules as well, they have multiple roles, right? They don't do one thing in the body, they do multiple things in the body. And that is why in the future, if you see a side effect, it's probably because it is reacting with the body in a different way, just because we can't do anything about it, because that reaction can happen in so many different parts of the body. So as I said, we will now move into the main part of the immune system that we will talk about at least, is the antibody, right? So again, big words, you don't have to read immunoglobulin, which is just a word, which is just a type of class of protein, right? But an antibody, well, as the name suggests, and I'm sure we've all learned it in school, what is it? Well, it is responsible for making sure that it keeps the body safe, right, from any outside object, right? Protein, like bacteria, virus, right? It is your antibodies in your body are responsible for protecting you from any of those agents. And we will get into it a little bit as to how it does it. But the main job of an antibody is to protect you from anything. Now, before at least, we always used to speak about antibodies as something that is to help us from foreign objects, right, like bacteria and viruses. But there are a lot of diseases that are caused by genetics, right? That we have in the body that causes like a genetic mutation that causes a disease. Antibodies are also helpful with diseases like those. And again, we'll talk about this in the next few slides. But an antibody is primarily the biggest and strongest part of our immune system, and they help us fight more diseases, and they're responsible almost for single-handedly for taking care of us and making sure that we don't continue to fall sick all the time. So what is an antibody, right? So we can think about this in multiple ways, and we can visualize it in different ways, right? So this is how the traditional structure would look like, right? There is most of this part is the same for the antibody. So you will see the green color, right? And these two blue colors, they are identical. So if our body has, let's say, 10,000 different types of antibodies, this will be the same for all the antibody, the circular region. Only this small region will change. And why this is important is that, again, we will come to it in the next slide. But if you were ever had to write about an antibody, right, how would you write it? Well, do you see this weird, complicated alphabets? And I'm saying that this is the same as this. What does that mean? Well, it's quite simple, actually. So each of these small grids, right, are, I said, antibodies are proteins, right? And proteins are made of amino acids, right, which are polypeptides. And polypeptides are made of peptides, right? And peptides are what? Amino acids, right? And amino acids are chemicals, right? Very simple. We showed, right? The COOH, which is the acid, and the NH2 that forms the amines. It's very simple. So each of this small bit, like this particular section, can have about 300 to 400 amino acids. And that is why you start seeing these numbers. So if you split them into two, each of these small boxes is made of amino acids. And each amino acid, we can give them a name. So Y, F, C, R, G, all this you see here is just the amino acid that is here. And that is the part of A, because A is, let's say you can say, is the left chain, and B is the right chain. So again, it is telling you exactly what amino acid is here. in each of these sections. So if you have to write, let's say, a chemical formula for an antibody, right? You can't write, yes, say CH2OOH, right? That is a smaller formula. If you have to write something for an antibody, it would be too big, right? It is impossible for us to write a chemical formula for something like an antibody, right? Because it has, millions of carbon atoms, millions of hydrogen atoms, millions of oxygen and sulfur atoms. So you cannot write a chemical formula the way we would write NaCl or NaOH or anything like that. So we'll have to write this. We'll have to write something like this, because now we're shortening the names of the amino acids down into one alphabet each. And so we're saying A part of it will have these many amino acids, B part of it will have these many amino acids. So if you ever see anything written like this, don't worry, right? It is just shortening the amino acids chemical formula into smaller alphabets and kind of showing the chemical formula in this way. And we will use that for research, right? To make sure that we know exactly what is there in every antibody part of it. So if you again go into a little bit more detail, right? Like I said, we don't need to know everything, but this part of it will remain constant. And this is what remains different. And for every antibody, this is the part of it where it attaches itself to an antigen. So if it is an internal disease causing vector, right, like a genetic mutation or some antigen in the body, or if it's a virus or a bacteria itself. So how does it bind? That I will come to in the next slide. But if we want to look at it visually, like an antibody, right, this is the way you would draw it easily. This is how they would actually look like. They're strands, right? So you see the green strand here, right? It goes from here to here. And in actual reality, you see the blue strand here. That is what they mean by the green strand here. Right? This is the constant region, right, where you see most of it coming together, as showed in the same picture below. And this is where the difference usually occurs for every antibody standard. Now, if you saw this in the microscope, right, the same Y shape, this is how they would start looking like, right? This is a sample of the blood. And you can see that there are different types, and you saw the chemical formula, some of them here, right? Different colors, and each of the colors are different chemicals. So the red, again, like I showed you, is an oxygen. The green might be the carbons. The yellow might be the sulfurs. So why I showed you 4 different ways of antibodies is that, number one, like the complexity of chemical reaction, right? Number 2, how many different ways can they be portrayed in, or can they be shown in as, and why is it that they play a complicated role? So if you see any of these pictures, any of these images, any of these chemical formula in your future, in textbooks or in notes, already know what it is. It is an antibody. It's very simple. So we spoke a lot about, yes, the constant region and this is the part that is different. But just trying to understand, right? How do you think that an antibody protects you? Just pure curiosity. Anybody have any answers on, we always talk about how our white blood cells and red blood cells produce antibodies. How do you think that they work? How do you think that once they are produced, how do they protect your body from diseases or anything like that? What way do they work in? How does it know, what bacteria to attack or what virus to attack? How does it work? Anybody have any ideas that how antibodies might work? So this is how they would usually work. Okay. All right. So any antigen, right? Anything that is, we're going to call this a bacteria for now. Okay. We're going to call this a bacteria, and any bacteria or any virus will have something on this outermost self, right, which can be recognized by our body, and we'll call that an antigen. Okay. So some people have, let's say, allergies, right? Allergies to particular plants, allergies to particular, seeds, like seasonal allergies, right? Why is it? Because that if that seed has something that looks like this, right, which is an antigen, our body thinks that it is something very harmful. Even if it is not harmful, if it has something that looks like this, it's called an antigen. Our body thinks that it is going to attack us. So that is why the antibody is shaped, like I said, most of it is constant, right? So this part of it is the same thing. This part of it is the same thing as well for most antibodies, but only this is different. That is how the antibody will recognize the antigen, right? So for example, in this case, you can see that it is square, right? So then it is able to sit right here. So this is the region where the antibody detects the antigen and reacts with the antigen. Now, if the antigen was round in color, if it was round in shape, right, it wouldn't be able to sit here, right? It wouldn't even be able to enter in here. So this antibody will not work against any other antigen. It will only work against this antigen, right? The next time, if it's a next virus or it's a next plant, maybe the antigen is circular in shape, or maybe the antigen is triangular in shape. So this can't fit. So we will have to find another antibody in the body, right, which has a triangular, kind of opening here so that it can sit in perfectly, right? And that is what is also shown in the next slide, right? So it is called the antigen antibody reaction. Let's see, like I said, different antigen binding site in the antibody, right? And each antibody can only work against one type of antigen, right? So it is like circular, semicircular here, more square, more round here, and more like pointed here. So this is how our body, when we talk about having different types of antibodies in the body, this is why we need different types, right? Not one doesn't work anymore, because each antigen or each disease will have different antigens and will have different microbes or bacteria. And so we need different antibodies for all of them individually to be able to cure and to be able to attack the antigen. So when the antibody attaches itself to the antigen, it becomes a complex, right? It becomes one complex, and they don't do anything anymore. So the antigen can only be free. The antigen can only cause a disease or a problem to you if it is freely flowing in the body. But if the antibody now fuses with the antigen and now it is together, then it cannot cause the body a problem anymore. So this is basically how an antibody usually protects the human body from diseases and from causing any issues. So how are these made, right? So I spoke about how, like I said, that, some of these molecules can be, created chemically in the lab, right? When I also said that this is too complicated to create in a lab, right? This is too complicated to just say, I will mix different amino acids and we will create a protein, right? You can do that if it was 6 amino acids or 10 amino acids, right? Small units. What if it is 1,000 or 100,000 or a million amino acids? So this is why you usually say that, it is a little bit usually more difficult to create an antibody. So small molecule, by that, what they usually say is chemically synthesized, right? Like I said, in the lab, it is easy to make. So for example, acetaminophen has this structure, relatively easier to make. But slowly as they become bigger, then it becomes much more complex. So what do they mean by biological molecule? Anything that is produced in the body, on the human body or any other organism's body is a biological molecule. So an example for this could be insulin, right? Insulin is something that we take for diabetes, right? And it is produced in the human body, right? But how do we extract it? How do we make sure that we extract insulin from some other living being's body and manufacture it in the lab so that we can dose it for human beings who have diabetes, right? So this is a biological molecule. It's already more complex, right? You can see that here that was just a grid of some small chemicals. As you move here, you realize it's much more dense, right? It's so many more atoms, so many more molecules put together. So it becomes much more complicated. All of a sudden, you start to make an antibody that this is heavier than this. It is bigger than this. This is much more complex. So just making it in the lab we can't do that anymore. It's not easy to just mix chemicals together. How different are they, right? This is how different they are. So aspirin, which is what we take for headaches, right, is a chemical drug. Okay, it's 180 Dalton. Dalton is what molecular weight. So that is how you measure the weight of a molecule, right? We say it's Dalton. Dalton, one hydrogen atom is 1 Dalton, right? So we just say it's 180 Dalton, which is quite small, right? Not too complicated to make. We have insulin, like I said, which is 5,900, which is still bigger than that, right? It is almost 50 times, about 30 to 40 times bigger than this, right? So that is a big molecule, already more complicated, right? Erythroprotein, it is what is one of the proteins that is in the body. It's also an egg. Again, much more complicated if you had to make it. But think about this, right? If you had to make a monoclonal antibody, right? Monoclonal just means that it is one type. Like I said, each type you can, when you want to make an antibody, you want to make one type, right? One type makes sure that it cures one disease. We don't want to make too many types at the same time, right? We want this and this to be the same. So by the monoclonal, they just means that both both sides of the antibody are the same thing, and they try to target the same disease necessarily. So you see how much bigger it is, right? It is almost 100,000 times bigger than the small molecule, right? And that is why, again, growing it becomes much, much more complex. So I have a question here. Have any of you gone to any factories, right? Have any of you gone to any factories where they make any chemicals, or they make anything that is, in powder form or pills or food or anything like that? And can someone tell me what is the container that they make it in? Is it a small flask? Is it a big, like, how do they make these chemicals? Not medicines, anything, any factory that you have gone to. Can you tell me, what factory it was and what containers that they usually make it in these factories? Like food, medicine, anything is fine. I just want to know. I have never gone to a factory. So I wanted to know if anyone else has gone. Has anyone um, seen it, how things like this are made? Have ideas on how they might be made in factories?
Food items are made in large quantities. And
Correct. So usually they are containers, right? Usually they are made in either like large, like reactors or containers or something like that. And even something like this, right, like aspirin, are small chemicals, and you can usually make them in like things like reactors, like glass vessels, steel vessels, things like that. So, but to make things like this, like I said, you cannot make them anymore. So how do you start making things like this? Well, like I said, antibody is produced within the body, right, of a human being, but it's also produced in the body of other like living organisms, right? So we can start that we have the luxury that we can start to produce it in another living organism and then use it from there. So how do we start? Well, number one, we start from mice. Okay. There are four or five different steps, and we can go through them all the time. Again, this is not the point of this is to not uh, mug this up or by heart this. I'm just trying to explain the process. Okay. If any of you have any questions, please stop me immediately. So the first step, right? What is the first step that happens? Well, we have a mouse. What you usually do is that, like I said, we inject it with a particular antigen. So from the last few slides, we again realized that antigen is one that actually causes the issue in the body or causes a disease, right? So if you inject the mice with the antigen, what happens? Well, the mice's body thinks that it is being attacked by the antigen, right? So it produces antibodies in its own body, right? So the B cells, which are one of the, again, as part of the cells or proteins in the immune system, they usually are the ones that produces the antibody. So you inject the mouse with the antigen, which is the disease causing vector. The mouse's body produces antibodies. And what you can do is that we can start taking these B cells, right? Once these antibodies are started being produced, we can now take the antibodies out of the mouse, right? Collect some of the antibodies, and then now we can say, well, that's a very, very small amount, right? That is not enough to make in a factory or give to a human, right? One mouse, it's a very, very small amount that is producing. Now, how do we make that larger? How do we grow that out into a larger group and batch so that we can start selling it to people? So once the mouse itself starts producing it, and then we extract it and we isolate it from its own body, we then take it outside the body and now start growing in the lab, right? We're not manufacturing it, right? We're just growing already what is already there. So how do you do that? So you have the isolated B cells, right? Now you can start seeing that this has many different antibodies, right? You can see the yellow, red, blue, green. Well, why is that? Because like I said, our body already has so many of these, but we just want one of them. We don't want all of them, right? We just want the one that caused the reaction for the antigen that we injected into the mouse. These are already there. These were already there in the mouse's body. We are just trying to get this one particular antibody. That is the one that we wanted to be able to cure the disease that we are interested in. So let's say that if when maybe, for example, if COVID-19, when we were looking at it, we would have injected the mouse with the COVID-19, the virus, and then we would have looked to see, which one of these antibodies is the one that the mouse produces in its own body to help with the disease. So once you take out all of them, you have cells in the body. It's called a myeloma. What it is, it's usually something called like a myeloma is a cell that does not die. It continues reproducing in the body all the time. It does not die, and in some cases that is bad, right? Because cancer is caused by multiple cells that does not die at all, that keeps producing itself on its own, right? So cancer is, well, a bad outcome of having myeloma. But in this particular case, we're going to use the fact that these cells don't die, and we're going to fuse them with the B cells. How is this useful? Because like I said, these cells don't die. They always just duplicate. Now, all of a sudden, when you fuse these two together, right, and these cells don't die, when these cells start multiplying, now this cell is going to have the antibody with it. So now we started with a small batch of antibody, right, from a mouse, right? We're fusing them with the cells that don't die, right? And when these fused cells, when they keep replicating, right, they produce more and more antibody for us in small plates. A fused one is called a hybridoma. Again, purely just called hybrid myeloma, right? And just because it is not, it's called hybridoma because it's hybrid myeloma, hybrid because it has another component to it, which a normal myeloma cell wouldn't have before. So they reproduce very quickly, and they divide. And so what you usually do is that now you try to isolate each one individually, right? So like I said, different colors, different antibodies. What we want, we just want one of them, right? We just want one of them. So now our responsibility is to select and screen and to isolate them and make sure that we only have like one of the antibodies that we are necessarily interested in. So what we try to do is that we have, one container having different chemical reactions and chemical media here. So what you usually do is that you put these into different containers, having different conditions. Some of them might have glucose, some of them might have sugars, some of them might have carbohydrates. So what you usually say is that each cell requires a different condition to grow in. We don't know, right? Our body or the mouse's body might have provided a very good condition for these cells to grow in. We don't know how to do it outside the body. So now we test multiple conditions saying that, for example, a 10% glucose, 80% nitrogen might be what is needed for these cells to grow. So we make sure that we grow it in a way that everything else dies, or everything else grows much slower, but the one that we want grows much faster. So now all of a sudden the one that we're interested in, it starts growing much faster. It starts growing much faster, and all these grow much slower. So in the beginning, when we started off, if we started off with 20% pink, 20% green, 20% purple, and 20% blue, now all of a sudden at this step, we try to grow it in a manner where that this all now drops down to 1% or 2% or 3%, and then this becomes 98%. So we have to grow them carefully and make sure that only the one that we want is growing. And when it starts growing, now we can start isolating them, right? We can make sure that we pick each cell individually, because we don't want complicated cells coming together, right? We want one protein, and we want each of them be taken individually out and moved to different stages. Why is that helpful? Because now all of a sudden, rather than having one cell growing multiple times, you have 96 or 100 different varieties of cells growing, So instead of doing it in one place, right, with all of them put together, because if you grew them all together in 96 different types, all of them would have also grown, right? So at this step, you eliminate everything else. You take out all the other 4 proteins, you leave one protein behind. And then when you have the one protein here, you grow it out in different containers, like 96 different containers that you have here. And all of a sudden we have so much more of the protein than when we started with, right? So we started with the mice, right? We got it from the mouse's body, right? We fused it with cells, and now we remove the cells that we didn't want, and then we start growing it out in more quantities. The last one is purifying it, right? So like I said, even how much ever you grow correctly, or how much ever you make sure that you are picking the right cells and doing it correctly, it still has some impurities, right? And remember, we spoke about how making sure that during drug manufacture and release, we have to make sure that we are producing the same product each time, and making sure that it is pure, so that it doesn't cause any side effects or any problems for the human body. So this is how you purify it. So let's say, for example, that the only thing that we're actually interested in is the red color, right? We don't care about any of the other things here. So what we do is that we use this process called chromatography, and I'm sure that you have heard of chromatography different, in different criteria and conditions. So how do you use chromatography for purifying proteins, right? Necessarily. So we pass it through a bed of beads. Okay, so each small bead will have particular shape, and all of them will have the shape of the protein that we want. So you see that the shape of these beads are all the same, and they're the shape of the protein that we want right here. So let's say that you have a mixture of 100 proteins. Okay, you put this into the beads. Now what happens is that all these circular and triangular things cannot go into these beads. So what happens? They flow straight through. So as you can see, only the red one can actually go into the spaces that we have in the beads here. Everything else, all the green and all the squares and all the triangles, they flow straight through because they cannot sit in here. Now, all of a sudden, when you suddenly realize that, oh, wait, only the red ones are left over, all these ones have flowed through. Now, what we can do is that rather than, just letting the medicine, letting the protein in, we can flush it, right, strongly with lots of water or anything with high speed, so that we can, make this jump out of that space that it's sitting in. Now, all of a sudden, we have removed all these things. So when we flush it with water or anything like that, only thing that comes out is the medicine, right? Only thing is to come out that is a protein that we want. So in this case, we have purified, right? We started off with some red, some purple, some yellow. Now we have taken all these things away, and finally, after when we flush it with water or anything like that, now all of a sudden we only have what we are interested in, and what not the other impurities or the other impure things, we don't have them anymore. So now all of a sudden we started off from a mouse, complicated mice, cells, proteins. All of a sudden now we have what we want, right? So it took 4 steps to be able to get to this particular stage, right? And if you think about this, I explained it to you in clinical trials, like research development, this takes about the research and development takes about 4 to 6 years, right? To be able to correctly do this, to be able to do all these steps correctly, right? Each step takes about 1 year, 1.5 years. How to make these beads correctly, what is the shape of the protein, what are the other impurities that there are there, how to flush it out correctly. So these are all processes that take a lot of time, right? So to simplify it and explain it, I think it's a little bit easy to do, but to develop it, it takes a lot of understanding of, biology, chemistry, making sure that we understand even lots of physics as well, making sure that we understand, how to design the correct beads, what size of beads do we need to create, how to flow it down correctly. So all complex process, right? And again, this is just telling you preferential binding, which is what necessarily you say, right? Preferentially bind. So it can't bind everything to the beads at the same time. Only it prefers one particular thing, and that is based off of the shape. So for in this case, these beads preferentially bind the red. Everything else flows through the column. And the last step, right? is manufacturing, right? We have the powder, we have the protein that we necessarily want. What do we do? Very simple. Now we have what it needs. Now it can go into a factory where it can go into, vials. We can, whatever process that we just did, we can make it and make sure that like large scales, right? So when I asked you, have you seen chemicals made or gone to a factory and seen how they produce it? Well, I am sure that these were large containers, right? Like, 2,000 liters, 10,000 liters, 50,000 liters. So for manufacture, we start now making them in large factories, right? Like I said, rather than making them in small containers, like I said, after research, after development, we're ready to manufacture it. And now it can be done in large scales, right? 10,000 liters or so. And then we can take these and say, well, now we can put it, we can put it into a package, we can put it into a bottle, and then we can clean it up, make sure that it just looks good, and then send it out into the stores, send it out to the hospitals, so that this medicine and this protein can actually reach the patient and help cure diseases. So these are the multiple steps involved in making sure that a protein starts from goes from the body of a mouse, that we discover exactly what's the right one. We grow it larger, we remove all the impurities, we purify it, and then we're like manufacture it, and then now release it to all our patients. Anybody has any questions until for this process based off of what I just said? The presentation, so that was the last what I had. We pretty much touched base on all the things that involved in the research, involved in the almost the manufacture from start to finish. Now it's just questions that any of you may have, any doubts, any concerns that any of you might have for me. That is all I had in terms of explaining whatever I thought that might be useful for all of you to know.